preclinical research, and it's Wendy, Galpern, and Dietrich, Haubenberger, and Wendy will go first.
So over the course of the next two talks, Dietrich and I are going to talk about how to be an effective consumer of preclinical research. What's the importance as a clinician to really pay attention to what's presented in animal studies? So I'll be mostly focusing on issues around the reproducibility, scientific rigor, and the transparency of reporting, and how that impacts your ability to be a good consumer of clinical research. And then Dietrich is going to talk about some of the preclinical requirements from a safety perspective, with particular emphasis on FDA requirements. I do want to disclose that I'm a full-time employee of Janssen, which is part of Johnson & Johnson, and I'm not going to be speaking about any unlabeled products during the course of this presentation. So I want to go through some of the challenges that have been faced when looking at preclinical data and trying to translate them into novel therapies, and then also some of the challenges that have been found in the literature with reproducibility of preclinical studies, and some of the issues that are thought to be affecting this, especially around the scientific rigor of the study design and of the study conduct, and also in terms of transparency and reporting. And then I'll mention some efforts to address these concerns about the preclinical data, which also affects your clinical studies, with a particular emphasis on how NIH is approaching this difficulty. So in thinking about the challenges when translating preclinical data into novel therapies, there's several different things to be thinking about. So I think if you pick up any newspaper, any magazine, you're going to see lots of headlines suggesting that the disease has been cured. If you were to look up Alzheimer's disease, you'd think that there's no need for any additional research based on the headlines. But then, of course, when you look more closely at these articles, you see that oftentimes these are in vitro studies or mouse studies, and you think based on what you're seeing is that the disease is cured, but really this is just a mouse that's been cured. And across multiple neurological diseases, I think many mice have been cured, yet we really don't have any therapies from a disease modification standpoint for many diseases. And so I think it's really important to think through what you're seeing in terms of the data and how that impacts your clinical study. I think one illustrative example is looking at Parkinson's disease. This is just a slide summarizing some of the studies that have been done in Parkinson's, looking at different disease-modifying therapies, and numerous drugs have been studied. And in the second column, you can see a variety of different mechanisms of action that have been proposed, and then several preclinical studies that have shown that these drugs are effective in the preclinical models. But for each and every one of these studies that have then gone on to a translation study into patients, the study has come up negative, and these studies don't show a positive benefit for disease modification. And I think just seeing the number here, and this is just a fraction of what there is for Parkinson's, and this applies also to Alzheimer's and many other disorders, many studies have been done, yet nothing seems to translate clinically. So what could be some of the reasons for this lack of translation? One of the issues may be the mechanism of the drug and the animal models that have been used. So thinking about the disease mechanism, oftentimes we don't know what the mechanism of a disease is. And so when it's studied in a model, is the model really relevant to the disease mechanism itself? For Parkinson's disease, oftentimes an MPTP mouse model is used, but that's unlikely to be what the cause of Parkinson's disease really is in terms of mitochondrial inhibition. Also, are we targeting the appropriate target in human disease? Targeting mitochondrial in the mice model may not be relevant for the human disease itself. Also, when looking at the animal models, oftentimes they don't recapitulate the human disease. When thinking of, again, something like Parkinson's or Alzheimer's disease, these diseases are progressive, and they progress over years and years and years, but with the mouse models, oftentimes it's an acute toxin injection, and that's then used to be a reflection of what's happening in the disease, which really may not be accurate. Also, we have to think about the pathology of the human disease when thinking again about Parkinson's disease. When looking at the mouse models, you could have a synuclein mouse model that doesn't recapitulate any disease features, nor are there the pathologic features of the disease. Again, showing that some of these animal models may have limitations that affect the ability to translate your finding from preclinical work into patients. Other reasons that drugs may have difficulty when going from the preclinical to clinical stage are things about the drug properties itself. So does the intervention actually get to the target? Specifically for neurological diseases, you'd be surprised to realize how many studies are done with drugs that later on are found really not to cross the blood-brain barrier. And obviously that's going to be a huge limitation, but in animal models where you're injecting directly into the brain, you're going to get different results than you're going to see in a human study where it has to penetrate the blood-brain barrier itself. Also important is to consider whether or not your drug engages the target. Is it engaging the target at a sufficient concentration to mediate an effect? Is it present at a biologically active form? And is the dosage that you're giving sufficient? Oftentimes you'll see a dose that's been tested in phase 3, but when you look back to see a phase 2 dose finding study and the rationale for moving forward a particular dose, there may not be sufficient data or a really good dose finding study to warrant the dose that was chosen. And Dietrich's going to talk a little bit more about properties of drugs in terms of thinking through what the toxicity is and how that will impact your translation, and also identification of the maximum tolerated dose to identify what your maximum dose moving forward should be. And additional reasons for difficulty in translating preclinical findings into the clinic is around reproducibility of studies. Oftentimes, studies are done in one lab, and then when people go to try to repeat them, they can't. Some of this may be due to the fact of the design of the preclinical study. Also, when looking at the preclinical data in a manuscript, there often aren't sufficient details to allow you to actually repeat the study that was done. And also, negative studies often aren't published, and so then there's just a lack of knowledge in the literature in terms of what findings may influence how you would make your decision to move the drug forward. So, as clinicians, is this really something that you need to care about? These are animal studies. How important are these preclinical data as you're making a decision to move the drug forward? So Shai Silverberg, who was at NINDS several years ago, wrote a paper about this particular issue, should clinicians care about preclinical animal research? And he emphasizes the fact that really you should. These are decisions that you're making to expose patients to new drugs, and you need to be sure that the data that are supporting this decision are strong. And I think that this is probably a nice paper just to review, to hear a perspective, to discuss some of the limitations of what are included in these preclinical papers and how that should be impacting your decisions. So I do want to spend a little bit of time talking about some of the challenges in terms of the reproducibility of preclinical studies and how the design of the study and the reporting impacts this difficulty. So I think this paper from Nature, again several years ago, 2012, really highlights the problem, and I was quite surprised when I initially came across this paper. So Amgen looked at 53 papers, and these were deemed landmark studies. And then when they tried to replicate these studies, they found that only 6 could be repeated, so only 11% of studies that were thought to be really profound advancements couldn't even be repeated. So again, highlighting the fact that there are difficulties just based on the preclinical data alone when we're trying to design clinical trials. So what factors contribute to this difficulty with reproducibility of studies? Oftentimes it may be that there's insufficient training of the researchers in terms of the experimental design that they should be using. Also, especially when trying to advance early on in a career or even later, there's a lot of emphasis on really trying to make a really strong argument about something that's provocative or new to have attention and advance the field forward. And this is actually a paper from Francis Collins at NIH, where they talk about an overinterpretation of hypothesis-generating experiments. So people want to be creative, they want to have something new, yet you have to be careful of how you interpret the data. And also, again, when looking at the details that are contained within manuscripts, there's often not sufficient information about the experimental design to allow an accurate repetition of the study. Other issues around preclinical studies that are important to consider is that, again, oftentimes negative data aren't published. So you see positive data, and that's all you're basing your information and decisions on, but then you may find out later on someone did try to repeat it and they couldn't, or they did another experiment that was negative that would have influenced your decision making if those are never published. Also, there's rare publications that talk about limitations or scientific flaws in other papers. And again, if data aren't published, you're not going to know, and these things really could impact your interpretation and finding. So again, from this paper by Francis Collins back in 2014, they identified 5 elements that were viewed to be critical for the design of studies. And these are relevant not only to clinical studies, these are issues we've talked about in our small groups, but certainly this extends beyond that, and they emphasize the importance of considering these elements in preclinical studies. So blinding of the investigator, randomization, replication of your study results to make sure what you've done yourself can be repeated, Sample size calculation. Why is it that someone did an 8-month study versus a 20-month study? This is rarely defined within the study. And then also looking at differences between males and females. Again, this is something that comes up not just in clinical studies, but now is being emphasized in preclinical studies as well. This is just a summary of what I think also is quite an interesting finding from 2006, looking at preclinical data. This is looking at 7 leading scientific journals like Science and Nature and Cell. They reviewed over 500 citations published between 1980 and 2000. And along the y-axis, you see different features that would be considered methodologically important to consider in an animal study. And then along the x-axis, you see percent. And you can see that some studies do include a dose response and they have long-term outcomes, but that's only around 60 or 70%. But as you go down this list, you'll see blinding less than 20%, adjustments for multiplicity, randomization. These are all rarely done in preclinical studies and something that really needs to be emphasized as you're looking at the preclinical studies, deciding to move forward into a clinical study based on these data. So what's been done to try to address these challenges? NIH has made significant efforts to really require in preclinical work identification of some key features which then extend into the clinical decision making. So if you were to Google anything about NIH, reproducibility, rigor, you'll come up with this webpage or something very similar, and it will have all the features that you really should be keeping in mind, especially for your grant applications. So NIH has identified 4 key areas of focus. One of them is the scientific premise. It's important from an NIH perspective, but also overall from any grant perspective or any decision perspective when moving forward with the study, is to consider the strengths and the weaknesses of the published data and look at the preliminary data that are supporting your decision to move forward. You want to take a close look at their experimental design and methods, make sure that it was rigorously done, and again, thinking back to that slide I showed you earlier, have they done the key elements that will make for a robust and reliable study? And then consideration of biological variables that should be factored both into the design and the analysis. And then also methods to make sure that the key features of the study are included in a paper so that people can actually repeat the study that was done. And when looking at clinical trial funding announcements, this isn't only something that applies to preclinical data. The clinical trial funding announcements also now expect that you're going to take into account the scientific rationale for your study and be able to defend it. So you need to be able to demonstrate that the experimental design was rigorous for the preclinical data that is supporting your clinical decision making, that strategies were used within those earlier studies to minimize bias, that the results are both robust and reproducible, and also to consider alternative interpretations of the data that have been presented. So back to the question from that paper I showed you a few moments ago in terms of a need for clinicians to care about preclinical animal research. I think the answer is yes, hopefully based on not only the fact that it needs to be in your grants, but also just highlighting some of the challenges. I hope that you now also agree that as clinicians, it is important to take a look at the preclinical data that are supporting your decision to move forward. And NIH does have a lot of information. Oftentimes their grants are difficult in terms of funding announcements to read through to understand what it is that you need to include. So again, if you just Google things like rigor and reproducibility, you can get summary charts that are going to really try to highlight for you what you need to include. And I think it's worth taking a look at those. I should mention these things get updated fairly frequently. They have notices and tables and all sorts of resources to help you, but do take a look to make sure when you apply that you go to the most recent announcement to make sure that nothing has been changed in terms of these notices that apply to the grant applications. So overall, in summary, hopefully you now do all recognize the limitations of preclinical data and the importance of the information that's contained in those studies to help inform the design of your clinical trials. There's been lots of efforts to improve the scientific rigor in these preclinical studies. There's requirements from journals now in terms of increased transparency and reporting to allow for studies to be reproduced. And also it's important as clinicians to take a step back when looking at the scientific findings. You want to ask yourself, has the study been designed well? Is it a rigorous study? Have they taken efforts to minimize bias? Have they been able to replicate the results, or have other teams replicated the results? And have the interpretations been looked at from different perspectives to make sure that these are the most valid conclusions?
Yeah, it's tough to talk about non-clinical things right after lunch, so I hope you guys can stay with us. I think these are all really important things that we sometimes don't think about, but we should think about, because overall, will increase our likelihood for success downstream. And I know that for a fact. Let me just try to close that down if I find the cursor. So my own story, sorry while I'm pulling that up here, is as I always say, I'm a child of this course myself. My study that I came to this course with 10 years ago now, 2009, was a study on using long-chain alcohols for the treatment of essential tremor. This was the idea of the project back then. And my study was the idea of a single ascending dose study, kind of a proof of principle, dose-finding study, looking at 3 different isomers of that compound, because we didn't really know which one was the best one, so we thought we were going to throw all 3 in and randomize them against placebo with a randomized sequence, etc. And we were all excited, submitted that to the FDA, and I was put on partial clinical hold, and I was only allowed to move forward with 1 compound because, as FDA told me, you did not show me that it is safe to go into human clinical trials with 2 out of those 3 compounds, which I thought, they're just long-chain alcohols. They are otherwise used as food supplements. Where is the problem? So that kind of opened my eye, and I went back and I did some reading that I should have done before, and eventually it let me focus a little bit on that topic, and I would have liked to have that talk given to myself back then, because I think these are really important elements, not only for the planning of your project to satisfy regulators. This is not what this is about. It's really about your compound, and how much you know about your compound that you're planning to study, and what you should know, and what you may not know now that you should know, which would help you to determine what is safe to go into subjects, not assuming that it's safe, but actually how can you prove it, and how, where can you get that information to convince yourself and your patients, and eventually also the regulators. So while we talk a lot about FDA requirements and getting approved or not, this is really about patient safety and about the success of your program. So I'm going to focus on the safety aspect of preclinical research and how that is relevant to you. Also, as a disclosure, I'm also industry employed with Neurocrine, but none of that talk is related to anything that that company or any of the compounds do. All right. So what are the kind of overall goals or questions that we want to have answered using preclinical safety studies? First of all, is it safe to put that drug candidate into humans? And if so, what would be a safe dose for human clinical trials? And by safe dose, we want to break it down a little bit. What would be the safe starting dose? You mitigate safety once you have determined a starting dose, and then you go into a dose escalation, for example. What would be a safe end dose, for example? And what are those limiting toxicities? So if there are toxicities, and often there are, you want to know about them, because you want to know what you should look for in your clinical trial. So this really drives your safety monitoring, your procedures in your trial. And also, are there any possible toxicities that you know of, that you learned from preclinical studies that are hard to monitor for in clinical trials? So histopathological changes, for example, so that are hard to monitor in the clinical trial. So you want to get some sort of a grasp about these aspects before you put your compounds into humans. If you then break it down to the individual studies that often are done and maybe required even to answer these questions. So the main goals and the principles of non-clinical testing is really to identify organ toxicity, to get an idea about the relationship of your safety finding that you see to drug exposure. If you want to determine, if you want to know about on and off target effects, if these are relevant to humans at all, and also that's also helpful then also for your planning of your study. Are there any good measures that you can use then also in humans to track and to monitor these potential safety findings such as biomarkers? these non-clinical safety studies that it's very likely that you yourself will be conducting as a PI, but that you will be relying on, or that you will be looking for, or that you will be asking, a manufacturer of a drug or a company that provides you the drug, you'll be asking for. These studies are, there's not like one set of studies that need to be done. It's really dependent on what type of therapeutic you are using. If it's a small molecule or biologic, these sometimes can be completely different. What's the therapeutic indication for CNS drug? You want to look at other aspects than for others. And also what's the scope and the design of your first in human trial, for example, treatment duration. So it's really about when you submit an IND, how is that IND opening protocol or that study, whether it's a single dose or multiple dose study, justified based on previous safety findings. So what should you know about your drug? And in that FDA IND breakout session, we talked about that a little bit. So one thing that you'll be submitting to the FDA when you send in your IND is that Form 1571. And that you have to sign as a sponsor of an IND, and with that comes a lot of responsibility. So when you check these boxes, and this FDA is really nicely laid out as a checkbox kind of procedure, the top part of the things that you have to do are pretty self-explanatory: table of contents, introductory statements, what's your current general investigational plan, protocol, etc. So you should have that already. But then there are 2 points here that I personally grappled with when I sent in my IND, and I just would not say made something up, but based on my best knowledge at that time from the literature and what I assumed, I just wrote basically 2 sections containing a few paragraphs on these sections: chemistry, manufacturing, and control, and pharmacotoxicology data. So this was a little bit of a black box for me, and I just sent it in, and then eventually that came then haunting me. And so what are these all about? So what does it mean, chemistry, manufacturing, and control? So a drug product and your substance, your compound that you will administer in your clinical trial is a drug product, right, at that point. So you would have to think about that also towards the FDA and towards also maybe your pharmacy will ask you about that, or even your IRB, what's the drug substance itself, the API, the active pharmaceutical ingredients, what are the excipients, what are the other compounds in your pill, that filler substances, flavoring substances, etc. What are impurities? Are there any impurities? You can assume that there are no impurities. Often, you have to have certain, there are certain tests to look for impurities. Even the container, what is it packaged in? Is this part of your drug product? So you have to think about that when you go into your trial. And you have to provide data on identity, strength, purity, and quality of the drug. And sometimes additional information such as manufacturer, storage information, and the stability. Usually, if you get a drug provided from a pharmaceutical company, you get all that information, or you get a letter of course reference to a file at FDA where all that information is already stored. So what about pharmacology and toxicology? So these studies look at the pharmacological effect and mechanisms in animals, and there's this acronym ADME that you might come across, and that stands for absorption, distribution, metabolism, and excretion. So think about it. Can you answer the question about your compound? How is it absorbed? How is it distributed? What's the metabolism? What's the excretion? These are all relevant points when you plan your study. For example, what are you going to do with subjects who have kidney function impairment? Are you going to exclude them, or do you have to monitor for that specifically, etc.? So what about toxicology? Toxicology can be acute toxicology. That's often sufficient for early stage trials where the administration is not that long. Subacute or chronic toxicology, depending on how long you plan to give the drug. What about certain safety pharmacology aspects of different systems? There might be specific studies necessary for cardiovascular safety or CNS safety, etc. Specific toxicology testing might be necessary if you have very specific routes of administration, transdermal, for example, intranasal. There might be, there are questions that you might need to answer about, irritability or so. Genetic toxicity might also be asked for, but that's often done in vitro. So that is overwhelming. So how many studies would have to be done until you're actually allowed to open up your IND? And then when you think about, here's in the pipeline, that yellow part here, this is where we are at home. Where we're good, where we're strong, we think that, get all that done before, and then, I'm ready to move forward. Often these studies are plentiful, and these studies are very, very expensive, and they are not all necessary, actually, to open up your R&D. So when you use the term preclinical, it kind of assumes that it's everything done that's before clinical, before you move into clinical. And actually, at the FDA, the review division, the head of toxicology reviewers slaps reviewers, clinical reviewers, literally on their hands when they use the term preclinical, because there's no such thing as preclinical. Preclinically, CMC testing or CMC data for phase 1 studies might just be necessary. It might be not just necessary to have an acute tox study done so you can open up your IND, so a short-term toxicology study to support a 12-week administration study, for example. There's lots of pre or better non-clinical stuff happening after you even start your clinical program, and for formulations, for example, chronic toxicology studies, you might have support to do 12 weeks. You might not have the support yet to do 6 months of administration, for example. Alternate, reproductive toxicology studies are usually done after, well underway in a clinical development program. So basically, there's no such thing as preclinical. It's a non-clinical development phase that sometimes goes even past marketing approval. So, what do you now need to do in your study as you get going? So if I stole some of these slides from FDA presenters, so that's often also how FDA takes a look at these things. So if you're using a drug that's off the shelf, approved by the FDA, and so you use an FDA approved drug off the shelf, so you can usually assume that if there's a label, that the drug product meets the standards for animal toxicology for maximum dose and the length of exposure as written in the label. So look up the label of drugs, and the way they write the label is very specific on what is allowed to use that drug for. So if you use that within the confines of that label, you should largely be fine. However, if you use a higher dose or a longer duration, different formulation, if you reformulate it, for example, or using a different route of administration than it's on the label, then potentially additional non-clinical studies or justification might be necessary. But you certainly have to think about if you give your approved drug, it's often what's happening, to a different patient population. That really then depends on the risk-benefit profile. So you might give a drug that's approved in cancer populations with a large number of toxicities to a more quote-unquote healthy population. That might be a problem because there's a different risk-benefit ratio there. So also, if you think about combining more than one approved drugs, FDA usually likes to know a little bit more about potential drug-drug interactions. So these are all things that you can think about. There's a way, we talked about that before. You can even ask questions to the FDA as part of a pre-IND interactions. Would that be necessary or not? Can you waive that? So that's something to think about. But if you stay within the confines of the label of an approved drug, often there's nothing else that you would need to do to go directly into your clinical trial. All right, so what about an investigational drug that is supplied by a different sponsor, like a drug that's not yet approved, but a different sponsor has under development or has an open IND at the FDA? Often the sponsor would not share with you the entirety of that IND paperwork, but they might be willing to send you a letter of cross-reference, which allows the FDA to go look into that often company's filed by the IND to look for support and justification for your study dose. And what you should ask that sponsor for is an investigator's brochure, and that's a document that the sponsor is legally required to always have up to date with all the studies that have been conducted, preclinical studies, also clinical studies if they have been, and that's a large, usually a thick document, and basically everything should be summarized to support your use of the IND in there. So the sponsor has to share that with you, and often you have to disclose that to your IRB. And again, all that data that's outlined in the investigator's brochure should support your planned dose and the duration and route of administration. What about if you use a dietary supplement? Dietary supplement, it's typically not an approved drug with an approved safe dose. And so you can't assume just because it's a dietary supplement that basically it has to be safe. If you use it, if it's used in the context of a clinical trial, then just by definition it's a drug. Even if the chemical substance is also used as as a dietary supplement, for example. So it has to adhere to the same requirements than other drugs. What if it's an investigational drug that you make yourself or your company pharmacy, for example? Then generally you must provide a full set of non-clinical pharmacology and tox data to support your IND opening study. How do you pick now your first study? Because let's say you have that information out of the investigator's brochure, or you even have some other studies available to you. You might not need anything additional if, again, there's an FDA-approved dosing range that's available, and that's typically written in the label. And if that's not the case, if there is data in the literature or any other study that you have at hand that support, again, dose range, duration of exposure, and the mode of administration. And that can come from animal studies, and even better, if there are human studies already out there that you can reference, they often can give a better evidence for the FDA to get approval. However, your reports, and even if you refer to the literature, they have to be quite specific in terms of how many animals or humans have been exposed, what were the doses, the duration, and the mode of administration. It always comes back to that. Ideally, obtain data sets. So if there's a publication that just says there were no adverse events, that's it, there's no table, and rather, probably have to go back to the office and say, would there be any chance that you can share that data set with you, where all the other grade 1s or smaller adverse or non-related adverse events are outlined, that actually where you actually have data for the FDA to go back to. Let's say you have your studies available, animal studies, for example, that tell you, that dose of 30 mg per kg, we're safe in dogs, for example. How do you now come to your actual human dose that you can use in your clinical trial? And there's a very nice guidance. And it says guidance for industry, and that threw me off. This is not just for industry, this is for basically every sponsor out there. FDA has a huge library of guidances, and there's this one guidance that actually helps you walk through that step. How do you come from animal studies to a dose that you can use in human studies? And that's outlined in 5 steps, and I will walk you through there. The first step that you have to look at when you read through an investigator's brochure, you go find the NOAEL, and that's the no observed adverse effect level. That's usually outlined in the investigator's brochure. And the definition of that is, this is the highest dose level that does not produce a significant increase in adverse effects in comparison to a control group. And these are usually adverse events that are biologically significant, and they're a benchmark for you to start converting to human doses. The second step, so once you have this dose out of animal studies, you would convert them to human equivalent doses. And this is largely true for small molecules. The nice thing about that is that toxic endpoints scale quite well between species when you normalize it by body surface area. And this table is copy-pasted from that FDA guidance, where you can actually look up, okay, so this was now let's say a rat study, and in that rat study, if I have a certain dose, the human equivalence dose would be divide the animal dose by 6.2. It's rather simple. So basically, it's a scaling by body surface area. Third step would be species selection. If you have a rat study and a dog study, which one do you consider? Usually, you want to go for animal models that are most predictive of human toxicities, and that really depends on how differences in metabolism, etc. Biologics leave out a little bit because they are sometimes different to assess here, because animal models and animals often don't express the relevant receptors or epitopes, so it's really focused on small molecules here. If you don't have any good idea about what is the most relevant species, you can always be safe and choose the species with the lowest human equivalent doses. Step 4, safety factor. Quite mechanistically, now you have your dose, your human equivalence dose, and now you divide it by 10. That's basically your that's your safety margin that FDA usually requires. There are reasons where you can argue for increasing or decreasing that factor. You might increase that factor if you have severe toxicities, for example, that show up at high doses in animals, or if you have non-monitorable toxicities. You might argue to decrease that safety factor if it's a well-known class of medication, or you have easily monitorable toxicities. But if you also do not know that, apply the factor of 10 and you should be good. Step 5, and the last one, is your pharmacologically active dose. So the pharmacologically active dose is the lowest dose tested in animal species with the intended pharmacological activity. So, and these are usually coming from pharmacodynamic models. And once you have your maximum recommended starting dose determined, you compare the human equivalence dose plus the safety factor to the pharmacologically active dose, and basically you would adjust your maximum recommended starting dose if your pharmacologically active dose is a lower one. So let's walk through an example. So let's say we have a non-clinical toxicology study that you pull out of your investigator's brochure, and they define an OL of 15 milligrams per kilograms in dogs, 50 in rats, and 50 in monkeys. So first step is, step number 2 is you convert it to human equivalence dose. You go back to the table, look up the numbers. So you divide the dog value by 1.8, the rat by 6.2, and the monkey you divide it by 3.1, and then you end up with human equivalence doses of 8, 8, and 60 milligrams per kilogram. You think about the equivalence and what is the most predictive model, and that would be in that case also the dog, and it's also the lower model, the lower equivalence dose. So the human equivalence dose that you move forward with would be 8 milligram per kilogram. You divide it by 10. And then your maximum recommended starting dose is 0.8 milligram per kilogram. So that's your dose that you can very well justify based on these studies to go into your clinical trials to submit to your IP. There are limitations, of course. This algorithm is quite mechanical. It's really focused on small molecules. Does not really apply to biologics. And there's a different approach now that you can look up in the literature. It's the minimum anticipated biological effect level, MABEL. The FDA loves acronyms. That's more looking at pharmacodynamic ranges compared to toxicology ranges. But still, if you lack all additional pharmacodynamic data, for example, this 5-step approach is still a very safe and very straightforward approach, and actually recommended by FDA to do. All right. What else can we read out of non-clinical safety studies? It's about clinical safety monitoring, and that really gives you an idea about your safety signals, what you should be looking for. But again, animals are not humans. You have to be vigilant about the unknowns. There's only 70% about concordance between safety signals seen from animal studies translating into human toxicity. So they are predictive to a certain extent, but there are about 30% of human toxicities that are not predicted. And this is a paper that I refer, and you can look that up then when we share the slides with you. There are certain adverse events that we see in clinical trials are often not predicted well out of animal studies. For example, here at the top would be the ones that only really show up in human studies: cramps, sweating, dry mouth, headache, dizziness, nausea. While at the bottom of that list are events where animal models might even be over-predictive: ataxia, convulsions, etc. Also, species-wise, the dog is really one of the higher predictive models compared to the mouse here on the other side. So that's often, in my case, where I was put on hold for 2 compounds. When then I looked into what would be necessary to do, they wanted to basically to do a 6 months dog study, which would have been financially and also time-wise, prohibitive. But the dog is a very predictive species here. So to summarize, in general, if human data is lacking, your non-clinical safety data is really important for your dose selection, for the planning of your safety monitoring procedures in your clinical trial, and also in the end to meet regulatory requirements. Human data is often more valuable than non-clinical data. So if you can pull that one study that has been done a few years ago, and you can show that these dose ranges have been administered without significant side effects, this will often mitigate for other drugs. That's eventually what my one compound that then eventually moved forward saved me because that one compound has been given to humans before, and I could show the dose ranges and exposures to humans that even in the absence of formal toxicology study convinced FDA it's going to be fine to give as a single dose in patients with tremor back then. Because non-clinical experiments, especially if you do them now with GLP, the GLP requirements are usually very expensive and time-consuming, and often are not covered by the typical grant mechanisms. I mean, there are grant mechanisms that if you get put on hold and if they require one particular non-clinical study, actually there are grant mechanisms at NIH where you can ask for funding for IND-enabling studies, as they are called. So we can talk about that as well. So it's not completely a red light if you get a hold letter based on lack of non-clinical information. Usually, you don't need to worry if your compound is FDA approved and you use it within the confines of a label. With that, I say thank you. With a nice picture from San Diego.